Hello students, welcome to Brilliant Qatar. In this video session, we are going to understand most complex topic from molecular base of inheritance, that is transcription in eukaryotes. In our previous videos, we discussed about the transcription in prokaryotes, right? But in this class, I am going to explain entire process of transcription in eukaryotes. Sir, is there a difference between this prokaryote transcription and eukaryote transcription? Yes. In eukaryotes, transcription process is not an easy process, students. It's a complex process. Lot of complicated things happen in the transcription of eukaryotes. So what are that process? How can mRNA form? Directly mRNA form from the DNA, or any intermediate stages are there? Any processing levels are there, or what? We are going to discuss. We are going to understand the entire process of transcription in eukaryotes. Look at here. If I take chromosome, look at here. If I take chromosome, in our last class, we have discussed this chromosome is made up of what? DNA, right? Look at here. This is chromosome. And in this entire chromosome, DNA is there. Right? Now this DNA part I am taking out. This DNA part I am taking out like this. Carefully look at here. This is a DNA part. In the DNA, there are two DNA strands are there. We know one is called template strand and one is called coding strand. Right? Now here, in this DNA strand, in this small DNA strand, I am taking this much of part is going to make RNA. Look at here, this much of part of DNA making RNA. So I can call this part is gene. And here, look at here, this one also. For example, this area also from here to here, this area also making a RNA. So I can call what? Gene. So what do you mean? The entire DNA does not going to make a mRNA? No, students. 2%, less than 2% of DNA only going to make a mRNA or RNA, we can say. So that part we called a gene. Look at here, this is a gene. I'm taking this part, a small segment which is going to give RNA like this. Which is going to give RNA. This part I'm calling gene. You understand my point? Sir, I have a question. Between these two genes, what is this area? Which not going to make RNA? What I should call this one? This is called the heterochromatin part. What is heterochromatin part? The DNA, the chromatin which is not participate in the formation of RNA. We call what? Heterochromatin part. So what is this area? This area is making mRNA. Yes, this mRNA finally translate into form what? Tell me. Protein. This is a central dogma, right? So this mRNA finally translate into form a protein in the cytoplasm. Yes or no, student? But I am going to explain now the transcription process in the eukaryotes. In eukaryote transcription, there are three steps are the same like a prokaryote transcription. Initiation, elongation, and termination. But whenever you talk about the eukaryote transcription, keep in mind, students, three different type of RNA polymerase enzymes involved in the transcription of eukaryotes. So what is this RNA polymerase enzyme? Do you remember in the last class I said, from the DNA, if RNA is going to form, we need one specific enzyme. What is this enzyme name? RNA polymerase enzyme. RNA polymerase enzyme. If this DNA is replicating to form a DNA, let me take here this DNA. For example, this DNA is replicating, this opening the replication 4, and if new DNA is forming, the complementary new DNA is forming, in this situation, which enzyme we need? DNA dependent DNA polymerase enzyme we need, because it is a replication process. So from the DNA, if new DNA is forming, that means DNA to DNA forming, we need DNA polymerase enzyme. But what is forming here? RNA is forming, right? From where? DNA. From DNA, RNA is forming. So which enzyme we need? DNA dependent RNA polymerase enzyme or RNA polymerase enzyme. Are you with me? You got my point. So, in the transcription process, which enzyme play a crucial role? RNA polymerase enzyme. But in case of prokaryotes, in prokaryotes, I told you single RNA polymerase enzyme can able to synthesize all type of what? RNAs. What are all type of RNAs? Now come here. All of you look at here. Before we go ahead, I wanted to explain the different type of RNAs first. RNAs. What are the different type of RNAs are participate in protein synthesis? 3 different type of RNAs are required for the synthesis of protein. What are the 3 different type of RNAs? Look at it. First RNA we call rRNA. rRNA. Second type of RNA we call, tell me, hnRNA. hnRNA, or this hnRNA undergo process to form what, tell me, mRNA. mRNA. Next RNA is called tRNA. tRNA. So there are 3 different type of RNAs are there. This 3 different type of RNA transcription need 3 different type of enzyme, students, in eukaryotes. In eukaryotes, for the transcription of 3 different type of RNAs, 3 different type of enzymes we require. Most important MCQ question, carefully listen. For the rRNA synthesis, for rRNA transcription, we need enzyme called RNA polymerase 1. RNA polymerase 1. RNA polymerase 1 enzyme is essential for the transcription of rRNA. Done. Now, what about this one? hnRNA and mRNA need, for the transcription of hnRNA and mRNA, we need RNA polymerase 2. RNA polymerase 2. For the transcription of tRNA, this tRNA, we need one more enzyme called RNA polymerase 3. RNA polymerase 3. How many different type of enzymes required for the synthesis of different type of RNAs? 3 in case of eukaryotes. But my dear students, keep in mind, if a question come, how many different type of RNA polymerase enzymes are required for the synthesis of all these 3 type of RNAs in prokaryotes, in bacteria? Your answer is 1 only. RNA polymerase only 1. That's why in NCERT clearly mentioned, single RNA polymerase enzyme have an ability to transcribe all 3 different type of RNAs in prokaryotes, in bacteria. In case of eukaryote, it's not like that. That's why I told you earlier at the beginning of this class what I said. Prokaryote transcription is not an easy process. Eukaryote transcription is not an easy process. It is little complex process. Several enzymes involved in this one. Eukaryotic transcription, complex process. Tell me, in eukaryotic transcription, how many RNAs? 3 RNAs. rRNA, hnRNA or mRNA, tRNA. For the transcription of these 3 different type of RNA, we need, answer me, 3 different type of enzymes. What are they? RNA polymerase 1 for rRNA synthesis, RNA polymerase 2 for hnRNA or mRNA, RNA polymerase 3 for tRNA. Most important thing, these rRNAs are 3 types again. MCQ, NEET question, carefully listen. One is called 28S rRNA. 28S rRNA. Second one is called 18S rRNA. Third one is called 5.8 sRNA. 5.8 sRNA. Remember, rRNA is also called abundant RNA in the cell. Abundant RNA MCQ question. Remember, in NEET like competitive purpose, this type of question come. Which of the following RNA is the abundant RNA? Which of the following RNA is the smallest RNA? tRNA is the smallest RNA. Small RNA, soluble RNA, adapter RNA, transferal RNA, hnRNA and mRNA, short-lived RNA. Remember all of you, short-lived RNA. You got my point? But in the tRNA also, students, in tRNA also there are important RNA is there, snRNA. What is this snRNA? Small nuclear RNA. Sir, what is the function of this snRNA? You come to know little later. I will explain you little later. So once you understand the different type of RNAs and different type of RNA polymerase enzyme, eukaryotic transcription process is easy now. Now shall we start? So we understood different type of RNAs, and for the different type of RNA synthesis, different type of enzymes. So I'm taking gene now. Directly I'm starting initiation process, elongation and termination process. Look at here. Directly I'm starting what? I'm taking one DNA sequence. Careful, look at here. This is DNA, gene. This is one gene sequence I'm taking. Right? Now here this prime I'm writing 3 prime, 5 prime, and here it is 5 prime and 3 prime. Students, done. This is a structural gene I'm taking. This is structural gene. We know out of the 2 DNA strand, 1 DNA strand only will make mRNA or RNA. Yes or no, student? The strand which is going to make RNA, we call it template strand, right? So which strand is that? 3 prime to 5 prime. This strand is called what? Tell me. Template strand. Template strand or non-coding strand. Non-coding strand. What about the other strand called? Can you guess what about the other strand called? Other strand is called coding strand or non-template strand. What is the second strand called? Tell me. This strand is called coding strand or non-template strand. Template strand. Remember all of you, 2 DNA strand never become what they call undergo transcription. They will never undergo transcription. If they undergo transcription, what will happen? We discussed in last class. Do you remember? If this 2 DNA strand undergo transcription, 2 different RNAs can form. The 2 RNAs are complementary each other, right? So they come and bind. dsRNA formation takes place. dsRNA never become protein. Do you remember this, students? So, out of the 2 DNA strand, 1 DNA strand only undergo what? Transcription. What is that strand called? Template strand. 3' to 5' strand, template strand. It is also called non-coding strand. Sir, why? Why this strand only? This could have also participated in the transcription, right? Why only this strand is participating? Students, keep in mind. Protein information is actually encrypted in the coding strand. To get that coding strand in mRNA, we need to transcribe the template strand. If you transcribe the template strand, kids, you will get coding strand in the mRNA. We don't want. If you transcribe the template strand, you will get the coding strand in the mRNA. We want that actually. Which strand we want actually? Coding strand we want. For example, if you transcribe the coding strand, what you will get? Template strand. We are not looking for the template strand, right? The information did not encrypted in the template strand, right? Where it's encrypted? Coding strand. So, which strand we should transcribe? Tell me. Template strand. Keep in mind, many students will get confused here. Which DNA strand undergo transcription? Template strand. Done? Now, careful, look at here. Here, before this, exactly look at here. Look at here. This is a gene part. This is entirely structural gene. Look at it. This entirely structural gene area. Done? So, before this structural gene area, there is a sequence is there. This sequence is called this sequence before structural gene. There is a specific sequence is there. This sequence is called promoter site, promoter region. Where is the promoter region? Just before the beginning of structural gene. Promoter site. Promoter site or promoter always attached from upstream to downstream. What is upstream? Upstream is 5' upstream. Upstream to downstream, 3rd prime. So promoter attaching always we will explain will reference with the coding strand. We will tell student. Look at the coding strand. So this is a 5' coding strand. So from 5' upstream to downstream. What attached? Tell me. Promoter site attached. Promoter. So I am representing promoter like this. Promoter site. Got my point? Promoter site from 5' to 3'. So with the reference of which DNA strand we will tell the promoter? Coding strand. Remember this strand. 5' to 3'. Promoter site. Right? Now careful, look at here. I told you we need an enzyme called what? RNA polymerase enzyme. In this process, we are making mRNA or hnRNA. First hnRNA will form and after that it convert into mRNA. For this process, can you tell me which enzyme required? Just now I told you RNA polymerase 2. Now let me take here. This is the enzyme. This enzyme is, I am writing here like this. RNA polymerase 2 enzyme. RNA polymerase 2 enzyme. RP2 enzyme I am representing. This RNA polymerase enzyme go and bind to promoter. This process called initiation. So this RNA polymerase 2 come and attach to, tell me, promoter. This process is called initiation of transcription. Initiation, step number 1. So this is step number 1. Let me take some what they call nucleotide sequence here, or let me represent like this. All are nucleotide. Okay, nucleotide sequence are there. I am not writing nucleotides here sequence here, but just assume this all are nucleotide sequence. What my point? Now, once this RNA polymerase enzyme come and attach to this promoter, it started to read what is in the template strand. And according to that one, it bring that RNA nucleotides and bind and started elongation process. Now here, look at here, careful look at here. From here, they started elongation process like this. New RNA will be start like this, synthesizing new RNA like this. Look at it, like this, till here. Now careful look at here. Which strand? 3' is 3' to 5' of DNA is undergoing transcription, right? Right? So that means new RNA. Tell me, here it is a complementary, right? So complementary means anti-parallel in nature. So this must be a 5'. 5'. So 5' towards 3'. That means, sir, what do you mean, sir? RNA synthesize always from 5' to 3'? Yes. Students, keep in mind, majority of students will do mistake here. 5' to 3' only RNA synthesis takes place. Even in DNA also. In DNA replication process, if you are able to recall, new DNA always grow from which prime to which prime? 5' to 3'. So here RNA also, which prime to which prime it is moving? Which prime to which prime it is synthesizing? 5' to 3'. Remember. Got my point? Understood? Now, at this RNA polymerase enzyme, sliding over, when RNA polymerase enzyme, look at the RNA polymerase enzyme 2, come and attached to a particular site. He said, this particular site, it is from 3' to 5'. This site is called terminator. This site I am writing here, this is called terminator. Remember. Where is the terminator? Downstream to upstream. Where is downstream? 3'. Look at here. This is 3'. Downstream. Downstream to this one. What is this one? This one called upstream. Most important NEET question. Promoter from which stream to which stream, my dear students? 5'. That means upstream to downstream. 5' to 3'. Then where is terminator? Terminator from 3' to 5'. That means downstream to upstream. Remember, this is the most important thing. So whenever terminator sequence come, RNA polymerase enzyme, look at here. Here the sequence is there. It is completely transcribing. This is called elongation process. Okay, and this continuously transcribing the new RNA and coming to this particular area. This is called terminator. Terminator say stop. Stop RNA polymerase enzyme. This is terminator. This terminator is, look at it. This is a terminator like this. Okay. This terminator is telling stop. Don't go beyond this one because the gene sequence still here only is there encrypted. After this one, beyond this one, don't need to go. So who is stopping? A particular area, particular sequence, terminator sequence. When RNA polymerase enzyme 2 come, it will be terminate the process of what? Transcription. What is step number 2? Step number 1 is initiation. Step number 2, look at this RNA polymerase enzyme started to synthesize the new nucleotide toward this 3'. So this process is called what? Tell me. Elongation. Elongation. Elongation is which step? Tell me. Step number 2. Once this RNA polymerase enzyme come to that sequence of terminator. Tell me what happened? The process of transcription will be terminated. So step number 3 is what? Termination. So termination. Step number 3. So tell me all of you students, in transcription process, 3 steps are there. What are they? Initiation, elongation, termination. Initiation. In initiation, RNA polymerase II enzyme combined bind to promoter sequence, and in elongation, it started to synthesize the new RNA. From which prime to which prime, student? 5' to 3'. Sir, is this our RNA? Is this our mRNA? No. No, it is not our mRNA. So what is this then? Now look at here. This process, whatever the RNA synthesized so far, look at here students. I am taking this RNA down. This RNA called hnRNA, heterogeneous nuclear RNA. Let's take down. Let me take this one down. Look at it. This one is hnRNA. I am representing like this. I am representing like this. It doesn't mean double strand, student. It is a single strand only. Just for our understanding, I am representing like this. It is a single strand only. It is not double strand. Now, this side is what? Tell me which prime is there? This is 5', right? 5', and this is what? 3'. Done. This RNA is called what? hnRNA. Sir, why it is called hnRNA? Listen, students. In this hnRNA, my dear kids, 2 portions are there. The portion which I am shading with this white, look at it, this one is called intron, exon. Look at it, this is the intron part. This part is called, this darkly shaded one is called intron part. And this lightly staining, this light part is called exon region. Again, this one is called what? Tell me, intron. And here that is exons are there. This one is exon are there. So hnRNA have exon, intron alternatively present. So 2 major portions are the regions are there. What are they? Exon and intron are there. That's why it's called heterogeneous nuclear RNA. Sir, what is this exon and intron? What is this exon and intron? Careful look at here. Careful look at it. Let me finish this one. I will explain you. Look at this. All are darkly shaded parts are called what? Tell me, introns. My dear kids, this darkly shaded intron parts are not going to make a protein. They are not coding for protein. Just assume. Careful listen here. Message is coming from where? DNA. Done. Let me explain with one small example. I am sending one message to one student. Just assume Diya. I am sending one message to a Diya, 12th grade student. Diya, particular time, tomorrow, particular day, particular time, particular chapter, you are going to have a class in Brilliant. Just I am sending the message. But when I'm sending the message, that time I'm mentioning, hi Diya, how are you? How's you doing? How's your studies going? What food you had today? That is all I'm mentioning here. This is the unnecessary part, right? So when I send a message, when the student receives the message, she will read this one. So she will ignore this all and she will receive the content. What is the exactly informations are going to give? Why did sir send the message? Tomorrow class is there. What time? Important one. Which day? Important one. Which topic? Important one. You got my point. So these are exon. Exon means what? The part which is going to convert into protein. Important one. What is intron? Hi Diya, how are you? How's you doing? These all are intron. That means nonsense. There's no need. So in a hnRNA, one thing is there. What is that? Coding, which is going to give a protein. Second one is called intron. No need. It's a waste part, non-coding part. It's not going to convert into protein. So answer me student, what is hnRNA? Heterogeneous nuclear RNA consists 2 regions. What are they? Intron, exon. We don't want what? Can you please answer me? We don't want what? Intron or exon? Sir, we don't need intron, right? Because its intron is not going to anyhow, it's not going to make a protein. You got my point? So now all of you careful look at here. This hnRNA undergo a process called splicing. Splicing. Chopping. Splicing. Now, careful look at here. I'm taking this region down like this. Careful look at here. This region I'm representing here. This is intron. Intron. You got my point? These all are introns. In between, what is there? Exon I'm taking. Exon. Exon. Okay? Careful look at here. Careful look at here. Do you remember in the last slide what I said? snRNA I said. Look at it. Go back. Go back this one. snRNA. The function of snRNA I'll tell you little later, student. I said this snRNA. Careful look at it. This snRNA. All of you look at here. Come and attach here. snRNA. Now let me write here. This is 5 dash. This is what? Tell me. 3 dash. 5 dash and 3 dash. Now what happened? This snRNA come and attach this total part. This total part. snRNA plus intron. This is intron, right? Intron. snRNA. This total part. Spliceosome we call. Spliceosome. Spliceosome. So this spliceosome, what they chop, they cut. You don't want it to go to that one. You will not, anyhow you're not going to make a protein, so you no need. Chalo, get out. So here also, this one, snRNA, okay, and intron. This entire part is what now going to chop. This one also, snRNA, and this entire one going to chop. I hope you all are following me. This process known as, this process called splicing. Which part is going to remove, student? Tell me. Intron part is going to remove. So what left? What left? Tell me. Exons only left, right? This all exons are going to join. Look at here. This all exon. Only what left? Tell me. Exon part only left, right? So this is the exon part. Right, white one is exon part. So all, remember all of you, only exon part. Now all exon is going to join. All exon going to join. Done. To this 5 prime. Toward this 5 prime. Remember all of you, toward this 5 prime, one unusual nucleotide combine add. It's called the capping. It's called what? Tell me. Capping. What is this cap? Look at it carefully. Toward this 5 prime, there is one. Look at it, all of you. One unusual cap-like structure come and add. This is called capping. Sir, what is this cap made up of? Right on. Let me take here. This cap made up of specific unusual nucleotide. What is that? Methylated guanosine triphosphate. What is this methylated guanosine triphosphate? Let's take here. We know in our last class we discussed the nucleotide, right? Look at here. This is a pentose sugar. This is a pentose sugar. Here guanine nitrogen base is there. Here carbon and phosphoric acid is there. Done. This is the second carbon, third carbon, like this. How many phosphates are there, student? One, right? So what I wrote here, triphosphate. That means 3 phosphate molecules are attached here. 3 phosphate molecules are there. Guanosine triphosphate. Sir, you wrote methylated guanosine. So to this G, guanine, what come and attach? Tell me. Methyl group attached. So this entire one is called what? Tell me. Methylated guanosine 1 nucleotide. Which nucleotide it is? Unusual nucleotide. Unusual nucleotide. MCQ question. Cap made up of what? Unusual nucleotide. What is that? Methylated guanosine. Where is that? At 5th prime, before start codon. Here there is a start codon is there, student. A, U, G. Before start codon, this prime. What is that? A cap is there. So capping. At which prime? 5 prime. What about this last 3rd prime? 3rd prime here, lot of adenines. A, many adenosine nucleotides will come and add. Adenylic acid. That means adenine nucleotides will come and add continuously. How many? 200 to 300. So 200 to 300 adenine come and attach to this 3 prime. So it is called tailing. Tailing. Remember all of tailing at 3 prime. Capping at 5 prime. You got my point? 5 prime of hnRNA, tailing at 3 prime of hnRNA. Are you with me, student? You got my point? Now, this, because of many adenines attached to 3 prime, this we are calling polyadenine. Polyadenine tail, we call polyadenine tail. This is the start one, and our last one is called what? Tell me. We know in the RNA last one is called stop codon is there, right? So after stop codon tail, before start codon, this is called start codon. Start codon. This one is called stop codon. So where is the cap? At 5 prime before start codon. You got my point? At 5 prime. Now here it is. After this stop codon, what is there? Adenine, polyadenine is there. This entire one is called mature mRNA. Mature messenger RNA. So hnRNA convert into matured mRNA. This process is called splicing. This splicing is not takes place in prokaryotes. Direct mRNA is coming from the DNA. But in case of eukaryote, this complicated process takes place. Capping, tailing, exon, intron remove, exon join. Tell me, where is this entire process takes place? Nucleus only. Sir, I have a question. This splicing process also takes place in nucleus? Yes. What come out from the nucleus? Fully matured, highly processed mRNA come out. Are you with me, student? You got my point. So this process called a transcription, formation of RNA from DNA. Which RNA forming? mRNA forming. In the mRNA, 3 nucleotides we call 1 codon. Look at it. 3 nucleotide 1 codon. This 3 nucleotide 1 codon is equal to 1 amino acid. This is a stop codon. Are you with me, student? You got my point. So this entire process in NCERT, look at here, the NCERT diagram is given here. The particular area, look at this particular area where transcription is going to start. Now, this is RNA polymerase enzyme, right? Look at that hnRNA come out. This hnRNA have exon, introns are there. Intron, non-coding part, exon, coding part is there, right? Now, RNA splicing, what happened? Tell me here. After this one, methylated guanosine triphosphate adding, polyadenine tail is adding. Finally, look at it. Here this entire intron part is going to be deleted, and finally, fully matured, what form? mRNA form. This process called what? This process called transcription. If you able to recall prokaryotic transcription, we did not discuss this one. In prokaryotic transcription, no splicing process, no intron removal, no exon joining, no capping, no tailing, nothing is there. You got my point, students? This is called what they call splicing. This splicing is a dominant. Splicing is representing dominant world of RNA. RNA dominant world. So this one, finally, this one will become mature mRNA. This mature mRNA undergoes translation process to form a proper protein. That process called translation. The process called translation. One more important thing. Sir, come back here. Sir, this RNA, where is this RNA present? rRNA. Where do we get this rRNA? Carefully look at here, student. This rRNA actually, if I take this is smaller subunit of ribosome, and this is larger subunit of ribosome. I'm taking larger subunit of ribosome. Student, keep in mind, these are ribosomes. How many of you remember in 11th grade, Cell is a Unit of Life chapter, we discussed this one. Ribosomes are made up of two things. What are they? rRNA. That means in the ribosome, there is a rRNA is there, rRNA, and some proteins also present. You know, 80 different type of proteins are there in ribosome. 80 different type of MCQ question, remember. This rRNA, largest RNA, is actually work like a catalyst, enzyme. What's the function of this rRNA? You know, this rRNA function is peptide bond formation. Peptide bond formation that we'll discuss in the translation process. So this is, even here also, this snRNA also work like an enzyme to cut, to chop what? Can you guess? To chop what? Intron. Because intron is what? Non-coding part. So look at here, this entire process is called, what, tell me? Translation process. Most important topic, lot of hidden MCQs are here. If you understand the entire theory, this concept, whatever the MCQ question come, you will answer, I am sure. Remember, which DNA strand will make RNA? Template strand, 3' to 5'. What is the sequence is present before the structural gene? Promoter. To this promoter sequence, which enzyme come and attach? RNA polymerase II enzyme come and attach. You understand my point? So how many steps are there in the transcription process? Initiation, elongation and termination. I hope this entire concept you understood very much. Thank you for watching this video.